Hi, everyone. Welcome. I'm Annabelle from BioBuzz, and we're super excited to have you guys all here for another one of our Career Catalyst sessions. So these are part of our Career Catalyst and Community Office Hours series. We do these every single Tuesday, and it's to help bring industry insiders to share real-world insights and actionable advice to help you move forward in your career. And today, we're going to be talking about visual storytelling in science. So kind of breaking down how to turn complex data into clear, compelling narratives. We're excited to welcome Afrida Rahman-Enyart from Tech. She's a senior marketing manager. She has dual PhDs in neuroscience and psychology. She did postdoctoral work at Northwestern and has extensive experience in scientific education and product strategy. And Afrida brings a unique perspective on designing the visuals that help to balance that accuracy with audience impact. Before we do get started and I hand over to Afrida, I do want to give a big thank you to our sponsor, BioTrack, for sponsoring today's session. So they have over 30 years of experience. They've trained over 17,000 scientists. But definitely go and check them out at biotrack.com because they are a great, great resource for upskilling. All right, let's get started. I'm going to hand it over to Afrida.
Awesome. Thank you so much, Annabelle, and thank you so much for this opportunity. I always love doing these sessions, and any session that I can do where I can kind of help the next generation of scientists and communicators. So let me go ahead and share my slides. Okay, great. So yeah, as Annabelle mentioned, today I'm going to be talking mainly on how you can kind of make, engaging visuals that kind of simplify complex scientific topics. And I'll be primarily focusing more so on kind of infographics, just because that's kind of the main experience that I have being in kind of the marketing aspect of things. I work quite a bit on, generating flyers and these types of graphics for those types of like educational materials. So that'll be my main focus. I do just want to highlight, I know Annabelle talked a little bit about my background, but I always like to go a little bit more in depth as kind of just like a way to show people like, how did I get to where I am, and kind of hopefully give you guys an idea of the trajectory that I took in case you guys have any questions for kind of the career development aspect of things. So I first started off when I was an undergrad, very adamant that I wanted to be a psychologist. So I did kind of all the necessary steps for going into, med school. I was a psychology major. And then my junior year of undergrad, I had joined a lab, and that's really where I fell in love with doing science. And so I kind of shifted gears, and I knew that instead of becoming a psychiatrist, now I wanted to go to grad school and kind of focus more on being a researcher. And so that's what I did. So I ended up going to Indiana University afterwards. There I focused more on molecular sciences. So when I was working in undergrad, I was in a clinical and cognitive lab, and then I kind of switched gears and decided that I wanted to kind of get more nitty gritty and do some more like neurobiology work. So I ended up getting dual PhDs in neuroscience and psychology. I was specifically working on a project that was studying developmental neurobiology. So I was looking at the neuregulin growth factors and their interaction with their receptors, the ERB receptors, and how they affected neuronal differentiation. After I was done with grad school, I admittedly was super burnt out. And so I really wanted to start looking for opportunities in industry. I started applying for industry positions, but had absolutely no luck. I think a lot of the blame was my resume. It just was not curated for an industry position. So I ended up doing a postdoc instead. So I went to Northwestern, did a postdoc in urology. So there I focused on studying gut dysbiosis and microglial activation in pelvic pain. And as you can see, that was kind of during the COVID era. So there was a lot of downtime where I wasn't in the lab. And so it gave me a lot of time to focus on what do I want to actually do? Where do I see myself? And to optimize things like my resume and focus more on kind of my interviewing skills. And so that's just what I did. I knew that I wanted to continue pursuing like a career in industry. So I focused a lot of my attention on optimizing those kind of materials to make me a better candidate. And then I ended up getting an interview at Protein Tech. I interviewed for the product manager position, but while I was interviewing, they decided that they actually wanted to hire me in as a scientific liaison instead. So I came in as a liaison, then added product management, and then very recently, I actually became the senior marketing manager. So that's kind of how I got to where I am today. So my job as the senior marketing manager is mainly to develop strategies for marketing our products, as well as kind of raising brand awareness. So I work quite a bit with our team to design things like campaigns and marketing materials. And I do just want to give an overview of Protein Tech really quickly in case you are unfamiliar with who we are. So Protein Tech in itself is mainly an antibody supplier. That's really what we're known for. That's how we started when we were founded in 2001. So because we've been kind of in the game for over 2 decades, we have a really expansive portfolio of antibodies. So we cover about 2/3 of the human proteome. We also offer immunoassays like ELISA kits and IHC kits. And then we also have things like antibody labeling kits and magnetic cell separation kits. Since then, we have expanded to incorporate other brands too. So we have our humankind branded cytokines and growth factors. So these are going to be proteins that you can use for cell culture, for like differentiation protocols, and they're going to be raised in human HEC293 cells. And we do this so that you can be confident that they have native folding, post-translational modifications, and glycosylation. And then we also have our Chromotech branded nanobody-based reagents. So nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity. So they're great for certain applications like immunoprecipitation or immunofluorescence, specifically if you're doing things like super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics, and they manufacture antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So with that, I can go ahead and kind of begin the content for today and why we're here, which is visual storytelling. So I want to start off with talking about what is the main goal and like the importance of scientific graphics and why we have them, right? So the main idea is going to be that you want to be able to communicate something that would be otherwise very complex to explain in words in a format that is easily digestible. So it's really there to clarify things like complex data, facilitate people's understanding and retention. So there's a better opportunity for you to remember something if it's in a visual depiction, because you can tend to kind of have that kind of photographic memory for a graphic more so than you can for, say, like a big chunk of text. It helps with cross-disciplinary communication, right? So even within the sciences, one scientist in one field might not necessarily know how to comprehend something from another science. So if you're doing things like collaborative work, which I'm sure everyone here is doing, it's a really great way of kind of explaining to people who might not be familiar with your particular field. And it also helps you improve your scientific impact, right? So if you incorporate graphics in publications, presentations, or even things like, grant submissions, right? So it allows people to kind of easily understand what the importance of your research is, so that they are able to kind of make a better judgment as to, their thoughts on it, whether this project is going to be funded, And it's going to give you kind of a better shot at kind of encompassing and incorporating the important ideas of your project. So when we are considering what makes an effective scientific visual, there are three main things that I always focus on. The number one thing, of course, is going to be clarity, right? So you want to ensure that you are minimizing any sort of clutter and highlighting the key message that you are wanting to portray. And I'll talk a little bit about, how do you clarify and minimize clutter without losing scientific accuracy? Because generally, whenever I talk to people about making these scientific visuals, that is one of the number one questions I get. It's like, how do you know what is the most important part of the message? So I'll talk a little bit about some tips for that too. So the second thing that you want to do is simplicity, right? So as I mentioned, show only what's necessary, what really highlights your message. And then number three is going to be, oops, sorry, it froze here. Consistency. So use aligned styles, colors, and labeling. So I kind of took this example here. This is from a blog, a protein tech blog on endothelial cells and their role in HIV progression. And so here the idea is to show how HIV kind of passes through the blood-brain barrier. So you can see a very clear title, Mechanisms for Blood-Brain Barrier Infiltration, and very clearly highlighting this is healthy, this is HIV positive. So you can see that there's kind of a loss of integrity here between the endothelial cells that allows for HIV to get in. And so you have that free virus entry, whereas here you have those tight junction proteins that are still maintaining the integrity of your blood-brain barrier. And so the HIV does not pass through. So this is a really great example of a scientific visual that shows clarity, simplicity, and consistency, right? So it all looks like it's one figure. It's very nicely using the same types of styles, colors, and labeling. And it's got only the key message and also kind of minimizing any sort of clutter, right? So a lot of other things are happening here, but to get the message across, we really only need to highlight certain cell types and certain biological processes that are occurring, and we can eliminate everything else. So I really do like this figure that we had incorporated. So as I mentioned, I'll talk about how do you make scientific content simple without oversimplifying, because you still want to make sure that you're getting that message across. So the first thing you want to do is you want to identify what is your main message. Why are you including this graphic? And that should be really the thing that is highlighted the most, right? So for example, with this graphic right here, what we're trying to highlight here is the workflow for generating intestinal organoids and then characterizing them. So that really should be the main message. And then any other information that you want to include can be included, but it should not be the key highlight, right? So here we've highlighted the cells that we really want to focus on. Everything else is in kind of like a beige type of color. And so everything else is kind of supporting material. Sorry, it is not progressing. 1 second. Let me see if I do that. Okay. So I pointed out this in the last bullet point. So highlighting what's important. So making sure that those are the things that pop out and are what you initially see when you look at the graphic. And then everything else is kind of muted. Using familiar symbols, colors, or shapes are going to help. So for example, using immune cells as circles, using mitochondria as orange ovals. You don't want someone to look at your graphic and try to decipher, what is this shape supposed to be? Is this supposed to be a cell? Is this supposed to be, a molecule? So you just want to make sure that you kind of use what is most generally used and what people might be familiar with. Avoid any sort of unnecessary decorative detail. So for example, although it looks nice, you don't want to go over the top with colors or gradients or even things like 3D effects. The only time you want to include these kind of more fancy type of decorations is if it's going to have some sort of purpose here. So if you're trying to symbolize that, there is a gradient in expression, in that situation, you might use like a color gradient. But other than that, you should try to keep to simple simple color scheme. And then consider how abstract you can get, right? So biology is one of those things, it's obviously very, very, very complicated, right? There's a lot of things going on, especially if you're looking at like signaling pathways. So you want to consider, what's the minimum that I can show, right? So can I show a receptor as a rectangle? So am I just showing like, hey, this ligand binds to this receptor and there's this cascade? Or do I need to show things like the domains, right? So am I showing, okay, so when this ligand binds to this receptor, there's dimerization, and so you see structural changes. So does the structure matter? So obviously, if you can get away with having the receptor as a rectangle, that is much easier to interpret than if you're trying to figure out how to kind of make it more of a like a structural receptor and kind of showcasing things like it dimerizing. So just thinking about how simple you can get with the art. Sometimes when you are generating an infographic, you might need to include a lot of information, and that's really okay. You just need to kind of be strategic with it, right? So this is a good example of an infographic that we have on our synaptic proteins flyer. So the synapse is very, very complicated. There's a lot of proteins and receptors that are involved. So here we're showing just that, so how kind of complex that synaptic, the postsynaptic density and presynaptic density can be. But we wanted to make sure that we included everything because this is on a flyer that highlights our antibodies for the synaptic proteins. And we have antibodies against all these proteins. So we want to make sure that we encompass that all in this graphic. So the main thing that you need to do when you have a lot of information that you're trying to convey is still have a main message. So here our message is synaptic proteins. You want to rely on clear labels. So everything is labeled, it's kind of color-coded, so people can very easily see what each protein corresponds to. And then having a neutral background with few colors to complement. So here the important things are the proteins. And so the actual, neuron is going to be in kind of this beige color, whereas everything else that we have antibodies for are going to be kind of popped out in different colors. And then consider things like, can you layer the information, right? So for example, if you're looking at, say, right here, you're looking at this AMPA receptor. So you want to have this overall picture, but you want to showcase more in detail kind of this signaling. Can you have an inset that kind of expands this to the side and shows it in more detail so that people can kind of look at that if they want to, or they can kind of look at this more general picture. Is it possible to break it up into multiple figures? So this is going to depend on what you're utilizing that graphic for. So if the graphic is going to be on, say, a social media post, maybe not. But if you're using a graphic for like a presentation, a poster, a flyer, you have the potential to break up your figure into multiple figures, or having things like part A, B, or C. And then can you use numbers to guide the audience? So for example, if you have a pathway that you're trying to show, can you have, the part where the ligand is binding to the receptor as number one, and kind of go down and guide people as to where they're supposed to look in your graphic, so that they know exactly the steps that they should be following. So how do you know if your graphic is effective? So the main thing that I personally do is I utilize my colleagues a lot. So anytime I design a new graphic, I will send it over to them and I will ask them, what am I trying to tell you here? Can you identify the key finding or the concept? Are there any details that you are absolutely, getting lost in? Um, and there are a lot of things that you'll find that you overlook, that you might think like, yeah, this is something that someone who knows this workflow might know. So for example, Protein Tech recently launched this product. It's called the blotting accelerator. It's basically a buffer that shortens the time that you need to do Western blot. So it shortens it to 40 minutes. And so I generated a graphic comparing, like, this is a traditional Western blot workflow. This is the Western blot workflow using the blotting accelerator. And then I sent it to my colleague and I was like, hey, how does this comparison figure look? And he comes back and he's like, it looks great, but you never mention where to put the accelerator. And I looked at it, I was like, oh my gosh, yeah, I don't ever talk about it. I just say, this is what it looks like if you use the accelerator, but I never put a step like add the accelerator here. See, there's just times where you just overlook things that could seem very obvious. And so it's always just important to get someone else's perspective on it. You also want to ask them things like, what is the first element that their eye is drawn to, right? So you want that element to be what you're trying to depict. Again, all my examples are going to be like flyers and stuff because this is my experience. But if I'm trying to showcase, our product compared to a competitor product, I want everyone's eyes to be drawn first to our product, and then they see, oh, okay, there's like comparison data here. Let me look at that data next. So that's going to be very important as well. Are they accurately interpreting the data or the concepts, or are details left out that change the interpretation? This is particularly the case when you're trying to summarize, complex information. So if you're trying to like summarize what is known about a topic, a lot of times, we can't summarize everything. So we might forget details here and there, or we might not want to include certain details, but you want that overall conclusion to be the same. So it's just important to make sure people are interpreting it correctly. And then is it accessible and readable? So you want to make sure that your text, if you have any text in your graphic, is large enough. It's using a readable font. You want to make sure that your graphic is colorblind friendly. So try to avoid things like, using red, green together. One of the things that I found is if, for example, a lot of IF images have kind of that red green, you can switch that red to like a magenta, and that's pretty colorblind friendly. And then the ultimate test is going to be if you have a legend for your graphic, can you cover that legend, look at that graphic, and be able to explain exactly what that graphic is showing? So for example, all the examples that I showed you today, none of those had legends, but I'm hoping that you guys were able to kind of know exactly what that graphic was trying to tell you within the time that I had it up on the slide. So I really quickly, we just want to go over some tools and resources for making graphics. I personally am really, really spoiled, so I don't make my own graphics. I usually use a graphic designer, but my team, I know uses BioRender and Canva quite a bit. There's also things like Illustrator, or you can even get away with using PowerPoint. PowerPoint has come a long way as well in terms of making graphics. But if you can, just using a graphic designer is great. Obviously, just because they have a lot of experience. So this is just an example of kind of what I generated for a flyer that we had. So I basically wanted to talk about neural stem cell differentiation and kind of the growth factors that are involved in the differentiation of these types of cells. And you can see this is what I sent to the graphic designer. And the graphic designer basically kept what I had drawn up, but just made it more pretty, made it kind of more muted colors, more of like what aligns with, Protein Tech branding. And then I do just want to mention that you don't necessarily have to reinvent the wheel, right? So obviously you don't want to copy someone else's graphic, but it's absolutely okay to, go onto Google and get some inspiration. A lot of times what I'll do is I'll find a bunch of different graphics and I'll create like a Frankenstein using all those different graphics and be like, send it to the graphic designer and be like, I want something that looks like this. But as I mentioned, BioRender also has a lot of these like capabilities. Canva also I know is very user friendly, and so it is something that I actually want to kind of learn a little bit more. But as I mentioned, there are just a lot of other tools that you can kind of incorporate. So just whatever fits best with your workflow. Is it? There it is. Okay. So some other considerations that you need to have when you're making a graphic is things like appropriate dimensions. So what is the purpose of your graphic? Are you posting it on social media? Are you using it for a poster, publication, a guide? In which case you need to kind of figure out how your material is going to be displayed. So obviously, if you're posting on something like Instagram, that's going to be much different than if you're making something for a poster. So just consider those dimensions. You also want to make sure that you limit things like your word count. So your graphic should not be overly wordy. So generally, the rule of thumb is making sure that graphic has less than 50 words. And then use symbols and icons that can save you on words to make your content engaging. So for example, this is a graphic that we used for a blog that was about writing a personal statement. Or this was, no, sorry, this was not the personal statement one. This is the one that was writing a literature review. And so here you can see in order to kind of make the graphic a little bit more engaging and to kind of save on some of the descriptor words, we incorporated some graphics that people are kind of familiar with in terms of workflows, and just to make it a little bit more engaging. And then I just want to end here and talk about some career resources that we have available at Protein Tech. So at Protein Tech, we are really, avid believers that we should be supporting our early career researchers. So we do have quite a few blogs, and we also have an early career researcher hub that has things like our upcoming, travel grants on there, interviews with other early career researchers. We have a lot of on-demand videos. So you can just scan this QR code and go onto our support tab, and you'll see all these resources available for you. So with that, I just wanted to mention that if you do have any questions after today, you can always email me. My email is afrida@ptglab.com. You can also connect with me on LinkedIn. So if you just scan this QR code, please do connect. And we do also host quite a few events on our platform as well. This includes things like, webinars. And we also have in-person events that we do. So if you are interested in learning about any of our events, do follow us on social media because we post about all our upcoming events there. So with that, I'm happy to take any questions that you guys might have.
All right, awesome. Thanks, Afrida. That was a great presentation. So now we're kind of going to move into our Q&A session here. So before I kind of open up the floor and you guys can ask those questions, two different ways. You can use that raise hand function at the bottom of the screen, or you can use the chat. I do want to start us off. So Frida, like as you kind of went through and talked through during your presentation, I noticed you were talking about some of the data visualization tools, and in general with the rise of AI, what do you kind of think about those tools that are coming up? I know a lot of people are talking about just how communication roles in general and science communication roles are changing. Have you used those, or people that work on your team use those? Do you see like limitations to it, or you guys trying to integrate it in any way?
Yeah, that's a great question. This is a discussion that we have quite a bit within our team. There's this inherent fear, I feel, of AI, taking over, AI taking people's jobs and things like that. I personally think that AI is it's not there yet. So I don't think there's anything to really worry about just yet. But I do think it's a great tool as kind of like a stepping stone, or for having kind of inspiration for things like graphics. So I personally, anytime I've asked AI to generate anything scientific, I'll look at it and I'll be like, this is wrong. Like this doesn't look great. But I will say that it's been good for giving me inspiration as how I want my graphic to look aesthetically. And so I've incorporated certain bits that I've generated from AI, but then kind of changed up the material. So I feel like when we're thinking about AI, we should less think about it as like an assistant and more so as like a tool, like how you would use a calculator or a computer or something like that, and drawing inspiration from there.
Definitely. Yeah, no, I think that's interesting you bring up that point too, because I definitely use it in my work, and I definitely tell people the same thing. I think of it as like a calculator. It's like it helps you like refine, can help you get ideas, but you definitely don't want to rely on it too much.
Yeah, for sure.
All right. So another question I have, when you're balancing that scientific accuracy with making those visuals more accessible to kind of like that broader audience, do you have any tips for kind of like trying to communicate with a non-scientific audience, if you're using visualizations or anything like that? Like, do you feel like you break it down too much, and then it sometimes loses that information, or do you have any tips kind of around that to sharing?
Yeah, so that's another good question. So in the current role that I'm in, a lot of the internal trainings that we do have to train people on our team that are not scientists. So we do create these kinds of visuals for non-scientists. And this is where I feel like a graphic designer does come in handy, because most oftentimes when you're working with a graphic designer, you can have a graphic designer that has some science experience, but the ones that we work with don't. So I utilize them quite a bit, where I explain something where I'm just like, you want this circle to be touching this circle, and this is what this means. And then I kind of ask for their interpretation. Like, does that come across? Or how do you think we should showcase this? So having a non-scientist on board while you're generating those graphics is going to be really, really helpful for you, because yeah, you don't want to lose the message, but you don't want to make it too complicated that they don't understand. So that would be my advice. Yeah.